Hello everyone, welcome to the St. Joseph's open web-based learning platform, that is how you learn. Also welcome to Department of Biotechnology, St. Joseph's College for Women, Autonomous, Visakhapatnam. The Department of Biotechnology presents the SMOOC, that is St. Joseph's massive open online course in plant and animal biotechnology. It's a free online modular 8-week course for life science background students. Will be offered at the UG level.
Welcome back to Plant and Animal Biotechnology course and the plant tissue culture techniques and second metabolite production. The chapter 6 applications of plant tissue culture, in this case somatic hybridization and embryogenesis, will be explained by Dr. A.V. Brahmam, HOD, Department of Biotechnology, St. Joseph College for Women, Autonomous, Visakhapatnam. Somatic hybridization and embryogenesis have the learning outcomes, the following learning outcomes. Students will be able to be aware with the idea of the in vitro process, evaluate the different isolation methods for protoplast and fusion and their culture for somatic hybrids, and factors involved in the somatic hybrids and culture of the protoplast, a mechanism, evaluating the mechanism of somatic embryogenesis, reproduce the protocols for pilot scale operations. First of all, protoplasts, the contents are in the somatic hybridization. Protoplasts must be isolated, fusioning of the protoplasts of desired species, identification and selection of somatic hybrids, culture and regeneration of hybrid cells, applications and future prospects. So these are the contents of the present chapter. First of all, somatic hybridization technique. This is the protocol for isolation of the somatic hybridization. That is somatic isolation of protoplasts, fusion of the protoplasts of desired species, identification and selection of somatic hybrid cells, culture of the hybrid cells, regeneration of the hybrid plantations. So first of all, the mechanical, we have two methods, that is mechanical method and enzymatic method. Involves the manual slicing the plant tissues, releases the protoplast, it is very less efficient. While in case of the enzymatic method, uses the enzymes like cellulase and pectinase, specific enzymes. If it is a cellulose associated plant tissues, can be degraded by the cellulase, or if it is a hard tissue pectinase, pectin, the pectin will be effectively degraded by the pectinase, so that's there is a less degradation and a less chances of the contamination also. They digest the cell walls, enzyme concentrations and incubation times must be optimized. So-called isolated protoplasts are plant cells without cell walls. Isolation affects the protoplasty, plant species, and enzyme purity also affects the isolation protocol. So mechanical method, plant tissue, first of all let us discuss the mechanical method. The plant tissue will be taken in a mortar and pestle. The cells will be plasmolized through mortar and pestle, and then take the aliquot from the suspension, observe under the microscope whether the cells are isolated or the walls are broken or not. Okay, and cut the cell wall with a knife. Generally here ultratome or microtomes can be used in advanced studies, and release the protoplasm. Whatever protoplasm is obtained, collect the protoplasm, and further studies will be used. In case of enzymatic method, leaf sterilization, that is hypochlorites will be used to remove of the epidermis with the scalpel and needle. Plasmolized cells will be subjected to either of the combination of the cellulase or a pectinase combination, or alone treatment can give rise to release of the protoplast. Protoplast will be released. Protoplast, if cellulose is still adherent to the plant cells, cellulase is the enzyme, as a unit of enzyme will be added to the suspension, and protoplast will be isolated. The isolated protoplast will be conserved in a mesotonic solutions like DMSO, dimethyl sulfoxide, or 10% sorbitol solutions, mannitol solutions can be used to preserve the isolated protoplast. Let us see the fusion of the protoplasts from the two different genomes. May be achieved through the spontaneous fusion, since they are once they are allowed in the suspension automatically, because of the fusing agent, they can automatically fuse themselves and forms the homokaryons and synkaryons. Induced fusion involves the chemical fusion, mechanical fusion, electrofusion, and intraspecific and intergenic fusions can be obtained from the spontaneous fusion of the protoplasts. So protoplasts fuse spontaneously during isolation process, mainly due to physical contact. Interspecific produce homokaryons. Best example, Cephalosporium acremonium. This technique offers the only way of carrying out crosses and genetic analysis. It's possible to produce a preliminary genetic map of 8 linkage groups for Cephalosporium acremonium genes, which enhance the production of antibiotics. So that is a very wonderful variety created through the protoplast fusion. Intergenic have no importance. Interspecific protoplast fusions are of much importance in the area where new products are to be produced. So due to new genetic setup, many novel secondary metabolites such as antibiotics may be utilized in order to control the contamination and optimized products of the protoplast. Best examples: Saccharomyces cerevisiae, Saccharomyces fermentale, Saccharomyces cerevisiae, Saccharomyces lipolytica, and Pseudomonas crassulaceae, Penicillium crassulaceae, and Penicillium notatum are the best varieties. The fusion can be achieved through chemical methods like high calcium concentration or polyethylene glycol mediated, and electrofusion. So these are the images taken of the tobacco plant protoplasts obtained by digesting away the cell wall or fused to produce a cell that acquired some of the characteristics of both the genetic backgrounds and can be regenerated into a plant with some traits from both the parental plants. This is the next picture, the B6B. Two tobacco plant protoplasts obtained by digesting away the cell wall or fused. Here in the first one, the cell walls or the protoplasts are adjusting themselves and attaching to each other and forms a single cell, a single protoplast. So let us see the viability, whether they are fused or not. Most frequently used staining method for assessing the protoplast viability: fluorescein diacetate staining process. Very good method, a dye that accumulate inside the plasma membrane of viable only. So this dye can evaluate only the live or viable protoplasts. So those protoplasts which are not fused or broken improperly may not taken up the FDA stain. Viable intact protoplast fluorescein yellow green within the 5 minutes, whether they are focused to the UV chamber. FDA is dissolved in sodium a ketone and used at a concentration of 0.1%. And the another staining, that is a phenosafranin staining, it is specific for dead protoplasts. In that way, the live and dead protoplasts can easily be separated, so that in this case phenosafranin that run red in staining procedure, viable cells remain unstained by phenosafranin because they may not accumulate, right? Since they are completely surrounded, because the dead ones are already broken, so that they allows the porosity also differs from live and dead protoplasts. And chalcofluor white staining binds to beta linked glycosides in the newly synthesized cell wall which is observed as a ring of fluorescence around the plasma membrane. So this is a marker test for the protoplast viability. So protoplasts have both maximum as well as minimum plastering densities for growth after fusion, and recommended to culture at a density of the 5 to 10 power 3 to 10 power 6 cells per ml is optimal size, optimal quantity to culture the protoplasts after fusion. And it has been observed that the 5 into 10 power 4 protoplasts per ml is the optimal size quantity to culture the protoplast. So these are the identification and the selection of the somatic hybrid cells. Hybrid identification based on difference between the parental cells and hybrid cells with respect to the pigmentation, cytoplasmic marker, and fluorescein thiocyanate and rhodamine thiocyanate are used for labeling hybrid cells. And the presence of chloroplasts, one of the good marker, visible marker, whether if it is there, they appear as a light greenish in color. Okay, nuclear staining. Heterokaryon is stained by carbon fusion acetocarmine or acetoacrin, acetoorsenstein, or the good choice to identify the fused hybrid cells. Let us see culture and regeneration of the hybrid cells. Once they are cultured, once they are fused and screened, identified, cultured on suitable medium or provided with the appropriate cultural condition, plants are induced to regenerate from hybrid calli. First of all, the callus is regenerated. Callus is an unorganized group of cells which can be established on MS medium supplemented with the auxin-rich medium. The protoplast regenerate a cell wall after callus is regenerated. The outer cells first regenerate the cell wall and go vigorous cell division and form callus. The callus can also be subcultured if it is required. Somatic embryogenesis begins from callus. When it is placed on nutrient medium lacking mannitol and auxin, the embryo develop into seedlings and finally mature plantations. So here, very specifically, somatic embryogenesis that begins once the callus is subcultured, secondary callus or tertiary callus. The callus is placed on the cytokinin-rich medium, especially kinetin, or as a natural cytokinin, or synthetic kinin IBA, 2IP, 2-isopentenyl adenine, and BAP, benzylaminopurin, can also support the growth and development of the further organogenesis from the calli. These hybrid plants must be at least partially fertile. They may not be appears to be 100% fertile because they need to withstand with the conditions provided in vitro conditions. In addition to having some useful property to be of any use in breeding schemes, these can be utilized. So this is a somatic embryogenesis schematic diagram. Cell and cleanup, initiation of the embryogenesis, induced cells, secondary medium, cultured, globular, heart-shaped, torpedo-shaped, and cotyledonary stage, and radicals will be formed from the torpedo stage. And the plantlets can be acclimatized in a shade net areas or in a greenhouse chamber. So these are some examples. Direct somatic embryogenesis. The plantlets can be raised from the embryoids, or indirect embryogenesis involving the callus phase. Here you can easily observe here the prominent callus, and then the greenish color appears to be synthesis, photosynthetic reactions that do occur. And see here, the naphthalinic acid plus 1 mg BAP can enhance the immature embryos on MS medium, and then the 2,4-D will be reduced and BAP will be enhanced to give rise to optimized sizes of the plant. So these are the applications as we already discussed in the previous chapter. Of course, the DNA will be, the naked DNA can be uptake to the protoplast. That is a more important significant aspect of the protoplast. And somatic hybrids will be produced in the present topic that deal they already completing combines the traits like a pomato. Diseases can be screened very easily through the protoplast culture. So variety of metabolic studies can be possible through the protoplast culture. And important somatic embryos, higher propagation rate if all the protocols are goes well and without contamination and suitable, very suitable in suspension culture. The agarized then solid agarized cultures, there are a lot of problems associated with the stratification or reddening of the media, contamination. Artificial seed production. Somatic embryos are very useful in producing artificial seeds. In present day, seed quality and quantity is not to the optimum mark. So that the fake seeds or inviable seeds are packaged and sold to the farmers, so the farmers are committing suicide. So in this way, the somatic embryogenesis gives the good platform for pilot scale production of the artificial seeds. Labor savings. Every time one skilled or a trained assistant is required to produce the somatic embryogenesis. So these are the references for the somatic embryogenesis. Thank you for your attention for the present topic, that is somatic embryogenesis. Thank you very much.